All right, I'll get going with this stuff. Good morning. I left you guys off with the green eye. We were so close to wrapping this chapter up. That's all good. So acanthamoeba we talked about. Again, we're thinking in this last little bit of stuff, eye involvement and a couple of different parasitic infections, so some eukaryotes, the amoeba here. And the next one is something a little bit disturbing to me as I see this eyeball being pried open. You can actually see a hint of a worm right within here. And then here's another image of it. The Loa worm is called the eye worm because that's where it likes to be. This is a type of roundworm or nematode. We'll talk about a handful of other nematodes as we progress through this last chunk of the semester. So loiasis is the actual infection from Loa, and there is all sorts of details that we're not really going to get bogged down with. The life cycles of worms are typically super complex. There are oftentimes a handful of hosts involved in the transmission, and then the adult stage of the worm, like we're seeing up here, we would call that, whoever harbors that would be the definitive host. So we happen to be a definitive host with this because we have the adult stage of these worms. They're transmitted by deer flies. As these flies bite us, they inoculate the parasite into us, and then it migrates through the body up into the eye and has the potential to cause blindness. So I think, and this basically wraps up this chapter. Like I said, we were so close. So acanthamoeba and loiasis, two of the ones we think about as far as some potential parasites for the eye. And I didn't put it in here. I thought about it last semester. I'm like, I should add this. And this morning, let me stop this share. Back to a new share. Where am I going? Right here. Let me share this. I want to share this one because I think this one's pretty impressive. Let me make that full screen so you're only seeing the actual slide I want to show you. Onchocerca volvulus causes river blindness. And I thought to myself, how can we talk about loiasis, the eye worm, and not talk about river blindness, another nematode infection, all right? So this Onchocerca volvulus is another roundworm. There are handfuls of these nematodes that can cause all sorts of infections. And in here, we're showing some other stuff. These nodules are called onchocercomas. That's what we're looking at right in here. This could also fit with skin manifestations from the first bit of this chapter with that, and then ultimately the blindness, which is kind of the end result with this parasite. So the adults are encapsulated under the skin and establish some pretty severe reactions as a result of it. And we kind of encase the worm as part of our own response to wall them off. The juveniles, juvenile stage of these worms can be in the eye, and they can attack the retina and ultimately lend to blindness. Individuals where these infections would be classified as endemic, you oftentimes see situations where the adults are severely infected with this and in the process of going blind. As we get older adults in this population, almost all of them are blind. And there are definitely instances in some areas in Africa where you see the children leading all of the adults as they're like holding on to ropes, leading them down to the river to get the water, to bring it back to the village, because every adult is blind and they're reliant on the children to help them get to the water. And the kicker is those kids are infected too. They're in the process. These are transmitted by black flies, which like rapidly flowing rivers or streams. So, oftentimes you think about stagnant water or something like that. Great for mosquitoes. Black flies, kind of the opposite, and they want rapidly flowing water. And so individuals close to these waterways have a greater likelihood of becoming infected. So I'm not going to go through any other details or show you any other slides with this. This is out of parasitology textbook. So stop sharing that and go back here. This and shifting gears now. It's all good. So this chapter is posted technically under next week's material, but next week I've got double down, so it's fitting that we get a jump start on it, I think. So moving into respiratory tract or respiratory system infections, there are a plethora of organisms that we can think about within this this body site. And, oftentimes, we're looking at all sorts of body sites as we move through here, but I think about some of the big players as we think about some infections and things that might be kind of more common to think about in a clinical situation. It also depends on where you're at. But in the United States, we think about lots of respiratory issues and GI issues. That's where we think about lots of potentials. And many of us probably can think of times that we might speculate that we got some food poisoning, potentially some bacteria that maybe caused some of these circumstances. All right. So respiratory and GI, there are numerous organisms, all sorts of different things. And the other body sites, not that there aren't a lot of things there. If you are thinking about the last chapter, you're like, wait a minute, there were lists of organisms, all right? Again, the tables, I think, are beneficial, all right, to help you through this. This is always kind of a disturbing image. Aerosol are produced in the process of coughing and sneezing, or just flat out talking. You're spewing out droplets of potentially, some infectious organisms that set the stage for that to be spread to others. Although we are trying to move away from some of this mask wearing idea, something like this brings it all back around, and you're like, slap a mask on, right? Let's control this stuff, right? So a little bit of anatomy as we move through here. We can break down the respiratory tract, starting with the upper respiratory tract, the ear, the mouth, the oral cavity fits within this upper tract, and then we will move down into the lungs with the lower respiratory tract. There are definitely differences when we think about the varied regions of the body. The upper respiratory tract has the potential for numerous organisms to colonize and be classified as normal flora. The lower respiratory tract, you would think not, right? That should be a sterile environment, and finding organisms in that habitat is a red flag and indicative of them being the culprit, all right? But thinking about the upper tract, there's a lot of stuff that we have to think about. Are these good guys? Are these bad guys? Where do they fit within this? And in some instances, those that are there as normal flora, if the stage is set, they could potentially be pathogenic. And some of these we think of maybe pathogenic elsewhere. Streptococcus pneumoniae that causes pneumonia can live and reside normally in your mouth. All right, a little bit of it in there, not that big of a deal. All right, but the potential for it to get into the lungs and colonize and result in pneumonia is an issue. All right, and potentially other circumstances set the stage that lends to it having that opportunity to take hold. Sometimes viral infections weaken and deplete your immune response, lending to secondary infections, maybe something bacterial that could possibly lend to this, all right? Oftentimes, and a test question talking about antibiotics, viruses aren't targeted by antimicrobial agents. Antibiotics don't work on virus, all right? But there are some times when you are infected with a virus and antibiotics are prescribed, and they're prescribed in a circumstance like I'm describing with the Streptococcus pneumoniae, as a mechanism to ensure that this infection doesn't open up the avenue for strep pneumo to come in and take hold and manifest as another situation. So that, although we think about that as maybe a no-no, we don't want to do that. There are some circumstances where they could be warranted to be prescribed in those cases. Okay, so I'm not going to go through and spout off all of the anatomy here. You can look through that and see those details if you are so inclined. As we move to the lower respiratory tract, as I said, typically thinking within this habitat or environment, a sterile situation. That doesn't mean things can't colonize and take hold, right? And we will go through details with that, but not something that we would say normally should be residing within there, all right? And all sorts of details, as far as where they may be colonizing, but within the alveoli, the air sacs, there are definitely some potentials and issues with infection in those circumstances. So we've got a cross section up here. We're looking at the lumen of the trachea and then goblet cells. Remember from, chapters back, we were showing some of these different cells, and the goblet cells are those that are capable of secreting copious amounts of mucus, right? So these goblet cells secreting mucus, there's also, if we kind of zoomed in more on them, we're pointing at them, but if we zoomed in more, this layer on the surface of these goblet cells is very fuzzy. It's full of all sorts of cilia on the tips of these cells that help to provide some motion to get that mucus moving or flowing through there, all right? So the pseudostratified columnar epithelial cells with the cilia on them, and then the goblet cells throughout there. So this is a mechanism. We could classify, the cilia and the mucus, part of that innate response, helping to prevent or hoping that we prevent organisms from getting in and being able to colonize. They may get stuck in the mucus, or they get pushed up and out of there with the motion of the cilia, but not always the case. So we showed with skin infections, the Streptococcus pyogenes as a potential for things. Well, there were handfuls of things, but one of the good gory pictures was the flesh-eating bacteria, the necrotizing fasciitis. I mentioned in that previous chapter, we oftentimes, or at least me, I oftentimes think about pyogenes as strep throat, as something that we would be thinking about for some infection in the oral cavity, not eating my leg off, all right? But that just depends on the circumstances, all right? So this image, this is an electron micrograph, but we can see the Streptococcus in these nice long chains. You can also see a few of these that are in the process of dividing, all right? Which I think is really pretty impressive as we look at that. So the chain's getting longer as we move through here. And There are handfuls of streptococci that we can think about, some of which might be good normal flora and others potentially pathogenic. The pyogenes, long chains, and the Streptococcus pneumoniae, we'll see it in a second, but I just talked about it. The Streptococcus pneumoniae, if we were to look at those guys and compare them to pyogenes, pneumoniae is two cocci together. We call them diplococci, not big long chains. All right. So if you looked at these two under a gram stain, they should look different. They're going to be gram-positive cocci, but their arrangement is different. All right. And then there's other tests we could do, oftentimes to differentiate pyogenes from pneumoniae. In a biochemical situation, we do some antimicrobial susceptibility tests and see which is which, and it helps to clue us in that way. We're not going to worry that much about that in here, all right? So the strep throat, streptococcal infections, we could see some localized pharyngitis, or it could be some systemic signs and symptoms as a potential scarlet fever with this rash that we're seeing is a possibility. And this we showed in the last chapter with some of the skin stuff, all right? So with strep throat, you see this nice red inflammation, lots of spots up in here called petechiae. This is where we've basically, the petechiae are these red spots where we've got some blood that's being, these cells that are basically losing blood. Pyogenes is a bacterium that we classify as beta hemolytic. So it's able to take those red blood cells and lyse them and destroy them. So in an environment like this and getting in and having access to that meal, if you will, causes these issues. It's basically in there feeding on some of that. You also potentially back toward the tonsils might see some whitish growth, lots of pus as a potential with that. There are a handful of things, all right? A pyogen, as Pyogenes is named, is a, or wait a minute, sorry. Pyrogen is a fever inducer. Pyogen is a pus producer, and it's got both of these things happening, all right? So when I think of pyrogen, I think of pyro, a pyromaniac and fire, temperature, right? So oftentimes with strep throat, we see elevated temperature, and you also can see this, although we're not seeing it here, we're showing the petechiae, the whitish growth in the back of the throat. There are numerous things we think about with this. It is important to recognize and treat. There are lots of things that can cause sore throats, and virus is a great one for that. But if it's bacterial, we truly want to know what's going on. And if it is streptococcus, it's important to treat with antibiotics to prevent issues like scarlet fever or other issues, glomerular nephritis, where there are immune complexes. That's a type 3 hypersensitivity reaction. Talked about that stuff before, where the immune complexes can cause some pretty serious consequences within the kidneys, right? So this also, this bacterium could be something that we classify as normal flora in your mouth, all right? It depends on the circumstances, all right? If it's there in low levels, held in check by other organisms, you may be all fine and good, all right? But there are potentials where it can take over and cause issues. With that said, if you've got it as kind of normal flora, no issues, we would say you are a carrier or a potential carrier, or you have that opportunity to possibly spread that to someone else, all right? So shifting gears the littlest bit to the ear, here we've got a couple of images, a healthy and an infected situation. So the first one with the healthy membrane, tympanic membrane, the eardrum, you can see some of the bones behind there that help to transmit the vibrations so you can perceive sound. All right. Here we've got an infection. There's all sorts of inflammation, redness. There's also some pus within here, and the membrane has ruptured or torn, all right, as a result of this chronic inflammation. And there are all sorts, we'll talk about some of that, all sorts of potentials with that. And, you know, oftentimes ear infections kind of a common occurrence in little kids. And it depends on the circumstances as far as what they're dealing with, how we may try to treat that. So looking at another individual's throat, in the last one with the streptococcus, you could partially see part of the uvula, or non-scientific, I call it the hangy down, right? Your uvula, in this case, is almost completely covered with this bacteria that creates what we call a pseudomembrane. Diphtheria, Corynebacterium diphtheriae is the culprit for diphtheria, creates these patches in the throat. Kind of leathery gray. It's a bunch of dead cells. There's pus and fibrin. There's some red cells and the bacteria too, all of it together, all right? Setting the stage for that, all right? And another instance of upper tract infections. As we move downward and think about some of the potentials for lower respiratory tract infections, there's all sorts of possibilities. We think about some things like pneumonia, not just Streptococcus pneumoniae. There are numerous organisms that can cause pulmonary issues, all right? So consolidations or lesions, as we see these darker bits in this radiograph, would be indicative of something, some sort of an organism within there. You can't see this radiograph and immediately say, oh, that's tuberculosis or it's streptococcus or whatever, although with some tuberculosis, those radiographs definitely would be a little more, would guide you down that path a little bit more. There are other things as well that might raise some flags with that. We'll hit some details with tuberculosis in a bit, all right? So electron micrograph of our Streptococcus pneumoniae, instead of those big long chains with the pyogenes, diplococci, and here's a couple more that are forming. Here's an actual gram stain. There's a bunch of other stuff within here. This is, I'm assuming, coming from a sputum specimen from the patient that was in turn cultivated and plated out. It says here from a blood culture. So the lancet-shaped diplococci, these little purple guys within here. And if we were to zoom in a little bit more on them or have a little bit better image, you could see the details of them kind of together. Really pretty prominent, all right? And definitely different than pyogenes if you were looking at that, all right? With that said, pyogenes could be in your mouth, pneumoniae could be in your mouth, both of which might be in there as normal flora, right? And so swabbing at the back of the throat and then plating it, growing it, and staining it up, you could definitely find either of those as a potential possibility. And dependent upon the individual, right? If you guys were in lab and swabbed the backs of your throats and cultivated things, my gut tells me you would see all sorts of different organisms, possibly pneumoniae or pyogenes. you would see all sorts of other organisms as well, gram positives, gram negatives, rods and cocci, all sorts of variation, all right? And then it depends on what's going on, all right? If you're rolling in there and you feel perfectly fine, you don't have a sore throat, you don't have a fever, you don't have any sorts of symptoms, and you swab your throat and see some of this, well, then you would really think these are just my bacteria, my normal flora, right? No real issues. But if it's the other way around and you've got some sore throat, you do have definite symptoms, and we cultivate this and see the pyogenies, right? Then the flag would be raised that that's really what's going on. If it was different and you've got more pulmonary issues, well, first off, you better not be coming to lab in the first place, right? But if you did, simply swabbing your mouth is not going to tell you you've got pneumonia, right? I mean, you might have it in your mouth, but it doesn't say that's what's causing this pulmonary issue. But a sputum specimen showing that within there would be indicative of that, all right? So it depends on what's up, where you're swabbing, and a little bit about the individual in the first place. Are they exhibiting any sorts of issues or not, all right? So this image is a plate that's called chocolate agar. I think maybe we've talked about chocolate agar once before. Chocolate agar is a specialized type of media. It's not chocolate. It's cooked blood agar. So they heat the blood. So when they make blood agar plates, they make the media kind of like nutrient agar. And then as that media is just about ready to pour into the plates, it's cooled down to about 50 or so degrees Celsius. They take some blood, oftentimes sheep blood, but there are other animals that we might use. Sheep is pretty common. They take the sheep blood, they add that to the media, swirl it around a little bit, and then they pour the plates. All right, you can't cook or autoclave the blood, or it does this, all right? It destroys those red cells, lyses them, and then all of the nutrients that were trapped inside of them are available for things that want them but can't get them on their own, all right? So it's a specialized type of media that helps to truly enhance some of these bacteria. And one of them that really loves this situation is Haemophilus. It needs these factors from the blood, but it can't destroy the blood on its own, all right? Factors like heme and NAD are substances that it wants, and this is how we can provide it, all right? So we've got a good image of these guys that look kind of glossy on here, kind of shiny. We talked about that last time. What's that shininess telling us? If you've got a bunch of colonies that look really glossy or glistening, any special details about that bacterium that it might have? Anybody tuning in from home that's listening? That's a capsule layer, right? The sugar coating, right? That these bacteria have. When we see this type of stuff, that indicates a virulence factor, a greater ability to cause some disease. If we can mask ourselves in a bunch of sugar, our immune cells, all they see is the sugar, right? And Haemophilus is a good one for that as well, all right? Haemophilus influenzae sounds like we're talking about the flu, influenza, right? But that's really, it's not the flu, right? That's a virus. But it does cause pulmonary issues. It causes pneumonia-like symptoms. There are other circumstances as well. We talked about pink eye, and Haemophilus is a culprit with that. But different idea as we're moving into the lungs. Here we've got an image of Mycoplasma. Mycoplasma pneumoniae is really kind of an oddball bacterium. These guys are, and I think we've described them early on. When we think about bacteria, we think about ideas like gram staining them to see what's up with their cell walls, gram positives and gram negatives, and we deal with them a little bit differently. Mycoplasma is different and it lacks a cell wall, all right? It's not, doesn't have a gram reaction because there's no peptidoglycan to pick up that stain in the first place. We need to look at it differently, all right? And some negative staining allows us to visualize it because there's no cell wall, right? They are something that we say are pleomorphic. They're not rods, they're not cockside, they can be whatever the heck they want, all right? They're shapeshifters, all right? Because there is no cell wall that says you are spherical, all right? So that's what we're pointing at within there, and they are very small compared to other bacteria. You guys have probably seen some E. coli or Pseudomonas. Both of those guys are gram-negative rods, little pink cells, all right? And you've seen some other stuff under the microscope that looked bigger, maybe some bacillus, big purple rods, all right? You've seen some bigger cells, and you've seen some cells that were really pretty small. Mycoplasma is smaller than those, all right? Super little, all right? Very difficult to visualize when you're trying to stain it up, all right? This is something, and this is coming, infected hamster, but this is something that when it plagues us causes pneumonia, but it's a little bit different than some of the pneumonias that we've mentioned or will be mentioning as we're progressing along. It's a little more mild. Mycoplasma pneumoniae causes pneumonia that we classify as walking pneumonia, all right? Something you've all probably heard about, all right? Pneumonia is typically pretty bad, all right? And if you get infected and have pneumonia, you oftentimes might find yourself in the hospital because you're seriously ill. But with walking pneumonia, it's a little bit different. You feel like you got a chest cold. You feel pretty bad, all right? But not so bad that you can't function, all right? And that's an issue because now you might be going about your business when you really should be resting, all right? And that's an issue, all right? So I kind of hope that as dealing with the pandemic has made us recognize some of these things, and you start feeling a little bit ill, whatever it might be, and you just stay home. Wouldn't that be better? Right? And I know deep down that's where 85 other students that should be in this classroom truly are, right? Still sleeping. That's what's happening. The 16 that apparently are tuning in, I bet they turned their computer on and went right back to sleep, right? Oh man, it's all silent. They're like, no. Oh, here's somebody chatting. They're like, I'm awake, right? Somebody tuned in at 8 o'clock Monday. I'm like, oh man, early, right? I don't believe it. All right. Oh, now look at all of them. They're like, shit, man, I'm listening, right? Sorry, excuse me. Let's see what you're saying. Ah, they're there.
What are they saying?
They're like, nope, we're here.
Okay.
Laughing. I doubt it. Okay. I believe it. You're there. Okay. 4 of you. So tuberculosis, this is a bacterium that it's, there's all sorts of ideas with how we might identify some of these things. New pathogens or re-emerging pathogens that set the stage for issues. And tuberculosis is something that we would classify as a re-emerging pathogen. TB has plagued us for forever, all right? There are all sorts of issues with tuberculosis and historical accounts going back to antiquity, all right? Talking about some of these issues and instances, all right? As same as its close cousin, leprosy, although it's not causing lung infections. All right. So with that, this bacterium has a handful of things that lend to its success. All right. The cell wall of these bacteria have some special substances in there called mycolic acid, a real wax-like substance that makes it difficult to stain unless you use some specialized staining techniques called acid-fast staining. That wax layer helps them to kind of like the sugar coating, gives them a little bit of a leg up with our immune response, but they take it a step further, all right? Tuberculosis can get phagocytized by macrophages. And as that macrophage, and in your lungs, they're called alveolar macrophages. These macrophages can engulf the tuberculosis bacterium. They pull it in. Now it's within a vesicle as it's been engulfed. But once it gets into that macrophage and it should be fusing, this vesicle fusing with a lysosome, it inhibits that and makes it so that it either doesn't fuse with the lysosome, or if it does, those digestive enzymes, those granules, don't do anything to it in the first place, all right? So it lives in the cell that should be killing it. It replicates and proliferates within that macrophage. It explodes it, releasing more tuberculosis bacteria to go on and infect other macrophages, all right? A brilliant way to survive by living inside of the cell that should be killing you. It's fascinating, and there's some other parasites that, or bacteria parasites too, that do that. They also can get walled off. Here we're seeing a fraction of the lung, the alveoli, the air sacs. We can form some granulomas, some tubercles around the bacteria, trying to encase them and prevent their proliferation. All right. So as that happens, and these are where we're trying to wall them off, this granuloma, we keep them in check, but that has the potential of opening up, rupturing, whatever, releasing these bacteria out. All right. So treatment with tuberculosis is super difficult because taking the antibiotics to try to combat them deals with the bacteria that are available, all right? But when they're hiding in a macrophage, you can't target them. And when they're walled off in a granuloma, you don't target them either. So they're still there, all right? And that sets the stage for prolonged treatment, trying to ensure that anytime this opens and lets them out, the drug is there to try to deal with it. That's a big problem, right? In the United States, tuberculosis happens, but it is considerably more rare than if you were to be traveling abroad. There are some areas where tuberculosis is a huge concern, and we would classify it as endemic, right? It's naturally present within that population, and other, like here, more sporadic, all right? So it truly depends. And there's pushing 2 billion people walking around on this planet that are infected with tuberculosis. It is disturbing to think about. And there are, with antimicrobial treatments and people not doing the course of treatment as they should, all sorts of issues have lent to multi-drug resistant strains, lending to problems that have compounded these issues considerably, all right? Which is pretty crazy. How does alcohol make TB worse? Does it? I don't know. Yeah, that's a good question. Does it? I'm seeing your question, Wiley, and I don't know if I have an answer for you. I will have to look into that. because off the top of my head, I do not know how it would make it worse. I mean, alcohol could thin your blood down, but I don't see how that has. You've heard it makes the symptoms worse. I don't know. I will look into that and get back to you. But again, off the top of my head, I don't know. We showed before with some of the immunology stuff, some of the TB testing, and we got a couple of images up here to show it again. This injecting a little bit of the tuberculin protein, it's a purified protein derivative. So it's a fraction of that bacterium, not something that is going to make you sick when it's administered in this situation. All right. You know, way back in the day, Robert Koch time, Koch and Pasteur doing all sorts of things and trying to figure out stuff. Robert Koch comes up with some of this stuff, studying tuberculosis and some of this protein, thinking of maybe a treatment, and it ultimately caused the demise of numerous individuals. Backing off the concentration makes it something that can be used as a diagnostic assay. And if over the course, so a delayed type reaction, 3 days or so, if it welts up, dependent upon, you know, the circumstances, what this could indicate that you are currently infected or that you have been exposed. It's not necessarily saying you have TB right now, but you could have mounted a response previously, and through a cell-mediated response, you're going to respond to this antigen accordingly. Okay, so quickly with this table, a handful of things that we have talked about and a few we have not. All right. I mentioned the tuberculosis as a re-emerging pathogen, something that we kind of thought, you know, antibiotics have, you know, put this to bed, but that's really not the case. Legionnaire's disease is different. It's something that we would really classify as a newly emerging pathogen, first really recognized to cause issues in the mid-1970s, all right? Not that it never caused disease before that, but there was a large outbreak that ultimately put it on the radar, all right? And numerous things in between, all right? So the ear infection, acute otis media, so we could see Haemophilus, strep pneumo, Moraxella, and some others that can cause ear infections. So possible effusion, there's fever, maybe, nausea and vomiting and diarrhea. It's oftentimes a secondary infection. So bacteria from the respiratory tract end up getting trapped in the eustachian tube, and it causes infection, all right? So some of these may be in your mouth as normal players that cause these problems. So sometimes treated with antimicrobial agents, sometimes not. Some of these bacteria, and Pseudomonas isn't listed up in here, but it could be, should be, probably. It's a good player for some of these ear infections, and it, along with some of these others, can form some biofilms, which make it really pretty difficult to treat with antimicrobials. The outer bit may be targeted, but deep inside, they're still viable. And that lends to them kind of re-emerging after you finish that course of antibiotics. A little bit later, you're right back into this mix. So as far as treatment goes, it kind of depends on the circumstances with that. So we've talked about those. Bordetella pertussis, we didn't mention, it's whooping cough or pertussis. Something that we think of as vaccination, same with diphtheria, the DTaP, diphtheria, tetanus, and pertussis are the DTaP. And regular childhood vaccine schedules helps to prevent these issues, all right? And not vaccinating sets the stage for potential fatal bacterial infections that we should be able to prevent, all right? Q fever is kind of an oddball. It was called this because they didn't know what caused it. Q for query. We're like, what is it? It's question mark, right? We don't know. Well, we now pretty have a firm understanding that Coxiella burnetii is our bacterium that causes Q fever. It just took some time to truly figure it out. So fevers, coughing, pneumonia, all sorts of issues, all of these, you know, except for the ear infection, we're thinking about stuff like that. And then we've talked strep and TB. Oops, excuse me, sorry, still a couple more. There's so many bacteria. Chlamydial pneumonia, chlamydophilia pneumoniae, cysticae, and trachomitis, all different species or genera up here. This is a little bit different. And it depends on some of the circumstances as far as what you might come in contact with. Respiratory droplets, aerosols from someone with the chlamydophilia pneumoniae. The cysticae would be exposure to bird feces. Pigeons are a pretty good one for this, all right? And then the last one would be exposure in the birth canal, can set the stage with that. We showed Haemophilus and the capsule layers that you can kind of see on that. So coughs and fever, low body temperature, fever or low body temperature, one way or the other. Maybe you get some of both, fever and then chills. Klebsiella pneumoniae is another one. I kind of wish I had a picture of him up there. That is a super good capsule producer, very mucoid colonies, all right, super snotty, all right. And it can cause some lung necrosis, so you can see bloody sputum, current jelly sputum, all right, which is like bright red sputum as a result of blood within there, all right. And it is definitely something that can be a fatal infection, all right. Klebsiella pneumoniae is something that is normally pretty resistant to antimicrobial agents, so it's hard to combat in the first place. I showed you mycoplasma, the pneumococcal pneumonia, pneumococcus, it's often called is Streptococcus pneumoniae, and there are some vaccines available for that dependent upon circumstances. And then our Pseudomonas down here at the bottom. It also, with that capsule layer, very viscous fluid or mucus, and another one that can be fatal. Pseudomonas and Klebsiella, both pretty drug resistant, right? Setting the stage for serious issues trying to combat them, right? To begin with. All right, guys, I'm gonna quit with that. We'll pick up with some viral stuff on Monday. Have a great weekend. For those of you tuning in at home, I was just joking. I know you're sort of paying attention, sort of. The four of you that told me. All right.